Okay, so got the record going.
Hello, my name is David Ingram. I am a consumer safety officer with the United States Food and Drug Administration. It is a great pleasure for me to be here to speak with you a little bit about our agency's perspectives on soil health and how soil health can directly contribute to the microbiological safety of many of the commodities that FDA now regulates. I work within a center in College
Hang on a
Park,
second here. I know that you aren't
Center
seeing
for
that.
Food Safety
Here,
and
where
Applied
the video
Nutrition,
is.
where
Okay, should be
a
seeing
group
it.
of us largely regard ourselves as the F in FDA. Together, we share a common mission to protect, promote, and protect the public health by
So
ensuring
if you can indicate in the chat box whether you're seeing the video, I appreciate it.
and honestly labeled. It's quite a large task, especially when you consider that FDA regulates over 80% of the food supply. Those commodities you see in red, as they are grown directly in soil, do directly reflect the quality of the soil, not only in terms of nutritional content, but potentially toxicological, microbiological, heavy metal safety. And we do have programs to handle all those contaminants. We'll talk mostly today about the microbiological safety, but if you are understanding more about our other programs for toxicological and chemical and pesticides, I encourage you to reach out. What you see here are the main pillars of probably, well, not probably, definitely one of the most groundbreaking pieces of legislation to affect food safety in the last 70 plus years, Food Safety Modernization Act. Here you see the main rules and regulations that truly does provide FDA a brand new set of tools to regulate foods from the farm all the way to your tables. Now, we do stop at your front door. We will not go inside your homes to understand the hazards associated with your cold chain storage in your fridge or the microbiological safety of your cutting boards, where let's face it, a lot of these pathogens do reside. We can talk more about that offline. But I'd like to bring the first highlighted topic here: standards for growing, harvesting, packing, and holding of produce for human consumption. This is commonly referred to as the produce safety rule. This does set minimum science-based standards for the control and reduction of many of the potential sources of contamination from the growing community, the growing fields, the production environment. So the produce safety rule is designed to reduce the inputs from each of these potential sources. And I will highlight today a little bit more about soil amendments, since that is the discussion of today. What you see here is a framework, and you can access this as well as the document called the Qualitative Assessment of Risk, which contains many of the science, much of the science associated with our rules and regulations, the produce safety rules, called the Qualitative Assessment of Risk. The link here is active. This is a very high level structure of how we package risk in terms of least risky to most risky. I don't think anyone would disagree that human waste is potentially the riskiest soil amendment that you can use to grow fresh fruits and vegetables. And on the opposite side of the spectrum, we have non-biological elemental chemical fertilizers, which are not without risk. But of course, all soil amendments have a potential to not only disseminate foodborne pathogens in the environment where produce are grown, but also enhance the survival and persistence of any residual pathogens that might already be in the growing environment. I expect some of the scientists will discuss those concepts later today as well. So what does FDA regulate in the produce safety rule? We regulate soil amendments of animal origin, and we have a whole subpart F, associated with how growers should approach soil amendments of animal origin. We have a new term for it. We're called BSAAO, biological soil amendments of animal origin. In subpart F, we regulate how these should be utilized for produce consumption, the production of produce. So in terms of treatment levels, treatments, we have scientifically validated treatment processes that we require certain documentation. If you would like to have 0 days to harvest restrictions on certain soil amendments of animal origin, we would expect those soil amendments to go through a scientifically validated treatment process. There are many treatment processes. There are heat, physical, chemical treatment processes. Any combination of these can provide a perfectly pasteurized and even microbiological sterile product if it's scientifically validated. Compost sits somewhere in the middle between an industrial process and raw manure. Of course, raw manure would be considered the most risky. If you're using raw manure and you are an organic grower, you're very familiar with the concept of application timing. The National Organic Program provides restrictions around not only how you can apply, so the application method would be, I don't believe you're allowed to surface apply raw manure. You need to till in your raw manure, but you must also wait 90 or 120 days after you apply that raw manure before you could harvest your crop. So the farther you can wait to harvest your crop after you apply raw manure, the safer that commodity is grown and will be. And that is a concept we are rapidly understanding a lot better now. Over the last 5 years, we have been working hard to characterize and understand foodborne pathogen survival and persistence in the growing environment when raw manure is used to fertilize a soil in order to maintain soil health and crop nutrient status. We're 5 years into a potentially 10-year program, and we're doing a risk assessment, which I'll talk about in a minute. But our efforts are designed to provide some sort of protection, which is currently reserved under the produce safety rule. We currently do not have any days to harvest restrictions on the use of raw manure. The only restrictions we do have for the use of raw manure is that you may not apply raw manure in a manner that directly contacts the commodities that you're growing. Until our risk assessment is complete, we say that until that time, when we do have a better handle on what provisions should be incorporated to maintain a level of safety that FDA is comfortable with for the production of commodities where raw manure is grown, we say that if you're following the National Organic Program 90 to 120 days to harvest, that is perfectly acceptable until further notice. So we haven't quite finished compiling all the data sets yet. Again, we're in year 5 of a potential 10-year program, but I'd like to highlight the progress we've made and what we're doing about it. Again, if you're making compost or you're buying compost, this product should not be considered a pathogen-free product, and compost must be utilized in accordance with the produce safety rule in terms of application method. You must apply compost in a manner that minimizes contact with your commodities. Compost cannot be blasted across an established bed of leafy greens because of the potential for residual pathogens in it. And we talk a lot more about this concept in our guidance to industry, which we'll talk a little bit about here. In our compliance and implementation guide, this is now draft form. We are, based on that much thanks to many of your comments, we're restructuring and providing examples that are more applicable to your operations. We have 6 steps in subpart F that we would like you to consider following in order to meet the minimum science-based standards, how to use raw manure and other soil amendments of animal origin safely. And again, I'd like to point out there's going to be a few changes, one of which I get my pointer working. Microbial standards for your treatment process. This is sort of new language. We do not I just want to highlight that we do not have microbiological standards for soil amendments. We have microbiological standards for treatment processes. And we talk a little bit more about this concept in the guidance. And I hope that the next version will make that a little more clear. So our risk assessment, what are we doing? We are conducting a series of experiments and collaborations with industry, academia, centers for excellence, and other government institutions such as USDA in order to understand one concept. That is, we're trying to quantify the risk of human illness associated with the consumption of produce when grown in fields where raw manure is used. So what does that mean? That means we need to get an understanding of what pathogens are currently in manure. So we have commissioned some work to understand the level and not only the presence, but the frequency with which you can recover Shiga toxigenic E. coli from dairy and cattle manure and salmonella from poultry manure. We've commissioned studies to understand, okay, once you know how many pathogens are in that manure, if you're applying that manure to different soil types and different geographical regions, what happens to the population over time? Good news is all populations do decline over time, but we need to understand and accurately capture and characterize those populations in order to understand what provisions might best be suited to implement in the produce safety rule. So once we understand what level of pathogens are available in the soil and to which the commodities are exposed, we need to understand just how well those soil particles to which potential pathogens might be attached can transfer to commodities in which they're grown based on irrigation events, rain events, not only to commodities that are grown above ground, such as the leafy greens you see here, but also stem fruits, which are a little higher, as well as root crops. We're working on this concept right now. And then, of course, the final stage, which we already have a handle on, is just how do pathogens persist in after harvest when the commodities go through post-harvest processing, potential washing, and cold chain storage, as well as in your supermarkets and your home. So just want to highlight briefly the players. I expect you'll have more discussions later today in the subsequent seminars, but I want to highlight our efforts here and the players, main players involved. Dr. Michelle J. Russell will be speaking later today. I know about some of her work, but our first step was to understand what population size and what frequency with which pathogens can persist in different manures. And here you see some of our activities to understand that. Probably one of the largest data sets ever contrived to understand pathogen persistence in soils was done by USDA's labs. Hats off to Dr. Milner and Dr. Sharma for conducting and orchestrating a huge study to collect thousands and thousands of data points in different geographical regions and to characterize pathogen persistence in different soils over time. You can see here many of the graphs that accurately characterize the pathogen persistence. Some of these pathogen populations go up and down and up and down. Some go up and down, some just go down. It's just absolute amazing how many different survival patterns you can find in different environments. So we were able to understand finally how different factors are associated, what factors are most important in persistence of pathogens through USDA's work. So thank you. And those data sets were crunched through the help with artificial intelligence. And this is very much in line with our new era in food safety. We're using machine learning to be able to take these data points and sets that were created by USDA in order to develop a model, a computer model that can accurately predict the survival patterns of these pathogens in different soils and different regions, based on rainfall, soil moisture, and many other concepts and factors that we have in our paper we published earlier this year. Highly recommend take a look at it. It's quite fascinating how we were able to do this. So now that we have an understanding of the pathogen persistence in soils and the population sizes through which in the manure that's added to soils, we're now at this third stage here where it's probably the most technically challenging to get transfer coefficients from the soil to the leaves. And so we're deep in this now for all root crops, low-growing crops, and stem fruits. And I just want to thank you for your time and efforts in this arena and interest in this arena and your attention today and learning more about what FDA does, how we're doing it, in order to understand how the safety of soils can affect the safety of the crops that we're growing. And not only that, but every foodborne outbreak we have has a negative effect on human nutrition in terms of consumer choices, commodities, recalls. It's just negative attention to certain crops is huge. So we're working hard every day to prevent foodborne outbreaks, to promote and protect public health, and also soil health is a major factor. So this is a rapidly growing area of science for us. We appreciate your time. If you have any science projects that you're interested in, we can leverage our science to help you understand. I have some web links there, and then of course every one of the good people on this slide are important to this effort that I'm presenting today. So I thank you for your time, and have a great rest of your day.
Okay, we'll go ahead and take a few questions at this point. I will share that David was not able to be with us this morning for the live Q&A, so maybe I can tap the shoulder of some of his colleagues that are on this morning, Laurel, Laura, Keith. Possibly some of these questions come in here, we can address them. So please go ahead and enter them into the chat box here, and as those come in, we'll try to address those. David was pulled out to another responsibility with FDA this morning. Got a message in the last 2 days of that. So the thing I would mention is that our list of speakers this morning actually is pretty much US wide. We've got folks from the East Coast, Southeast, all the way to the West Coast. So really glad to have such a diverse group, geographically speaking, but also in David's slides, many of the people there that he referred to as experts in the field are on our panel this morning. So, okay, well, don't see any questions coming in at this point. So again, use the chat box. If you think of something during the subsequent talks, go ahead and put it in the chat box and we'll try to address it. So go ahead and move on to the second presentation this morning. It's by Manan Sharma. The title is Persistence of Pathogens in Soil and Surveillance of Pathogens in Water. So we'll boot that one up at this point.
Okay, well, good morning or good afternoon. My name is Manan Sharma. I'm a research microbiologist at USDA Agricultural Research Service. I work in the Environmental Microbial and Food Safety Laboratory in Beltsville, Maryland. I'd just like to thank the organizers and the conveners of this session and this meeting for inviting me to speak today to talk a little bit about persistence of pathogens in soil and in irrigation water. Following up on what Dr. Ibram led off with, and some of the regulations around it. Today, we're going to just talk about how some of these things get into the pre-harvest produce environment, like is what we like to call it. And essentially, what we're talking about is where we know that we're using inputs that contain bacterial, parasitic, and viral pathogens. And what we want to do is utilize the nutrients and the natural resources to grow produce and to recycle some of these nutrients so they don't wind up in the environment and causing deleterious effects. We want to use those nutrients, but we want to leave the pathogens behind. Now we know that manure and irrigation water can contain many different bacterial, viral, and parasitic pathogens. Today we're just going to focus on the bacterial ones, but that's just my expertise, and the parasitic ones can also be important. When we're talking about bacteria on produce in pre-harvest environments, we're talking about bacteria that are not growing in a mammalian gut. They're not in warm-blooded environments. They don't have access to nutrients. They're subject to a lot of different stresses like sunlight, microbial competition in the soil, which isn't the same in the gut. There's a lot of ultraviolet radiation from the sunlight. It's a lot more dry than they're used to in soils without rain. The dynamic temperatures between days and nights, so cooler and hotter seasons, are not what they're used to. So this is a lot of physiological stress for them to survive in. And we have the saying that all produce safety is local because it's dependent on the soil and the type of crops you grow and the weather and what type of manure you might use. But a more technical way to say that is all pre-harvest produce safety is affected by the interaction of spatial, temporal, agricultural, and weather factors. Because it's really hard to dismiss the agricultural effects and sort of the location and the weather effects. We know we have a lot of different sources of surface water contamination. This is a slide I got from the EPA. And we're probably going to focus more on the cropland and the animal feedlots in this picture here, but we know that rural homes, city streets, suburban developments, these are all can be sources of various pollutants into the waterway. And when we have flooding events like we had last year in Iowa, and in the Midwest, and that can cause a lot of contamination as well, spreading these pathogens from one field to another or even into the waterways. So there's a lot of different ways to get things into water and potentially onto all the food that we eat. And the thing about tracking bacterial pathogens in pre-harvest environments is like these pathogens, when there's an outbreak, they don't leave like they don't leave a confession note, okay? They don't say was here in irrigation water after being in animal manure, and then we were transferred to crops that were consumed raw, and that's what caused this outbreak of infection. You're doing a lot of investigative microbiology here, and you're trying to backtrack to figure out, oh, it was here, where do we think it was before, and how did it get on this lettuce that was contaminated, or the cantaloupe that was contaminated? So there's really not ever a clear picture. There's not ever a really nice scorecard that shows going from point A to B to C to D to E to F. It's a lot of finding and then putting the pieces together. And we know in previous outbreaks that RV septic tanks have been a presumed source of bacterial pathogens to contaminate lettuce. We know that concentrated animal feeding operations have also been presumed sources of pathogens, and in both cases it was presumed that these sources contaminated irrigation water that then subsequently contaminated growing crops. And for example, with the CAFO, what you could have is two different mechanisms of contamination. You could have manure that actually runs off into irrigation water and contaminates the water and gets transferred to the crop, or you could have a direct transfer mechanism where there's manure dust that's generated and then transferred to the crop, manure dust generated here, and then transferred to the growing crop here through sort of a more direct transfer. For some work done in our lab, we think that the water as a vector, the water is just transporting this pathogen from A to B. But some work done in our lab and some other work done at the University of Vermont with Deb Near's group basically showed that these pathogens have the ability to grow in simulated manure runoff. And this was runoff that we created, that we simulated using a very common biological soil amendment of animal origin that Dr. Ingram talked about. These are heat-treated poultry pellets. This is a common common fertilizer that organic growers use. We basically made some simulated runoff with this, inoculated it with the pathogen Salmonella Newport. And if you look at our chart here, the blue lines is our runoff that's made with the heat-treated poultry pellets. The orange line is just soil with none of those poultry pellets added. And you can see that the Salmonella can actually grow to a much higher level in the amended runoff, the runoff that contains the poultry pellets, than in just the soil alone. So this is one way that once it gets into the water, if the conditions are right and the temperature is right, you can have growth of these pathogens. Some other work done in our lab by Ruth Odie, who now works for the FDA, but she evaluated the survival of Salmonella on spinach plants if they were introduced basically in dust particles or if they were introduced in water particles. And this red line is the Salmonella survival in the dust on the spinach leaf. The black lines are the Salmonella survival in the water on the spinach leaves. And what you can see pretty clearly from this graph is that the decline of the Salmonella in the dust is much, much slower and much smaller than it is for the Salmonella in the water. So what this indicates is that if you have that dust that's transferred and has a pathogen in it, those pathogens can survive for a much longer duration than if it's actually in water. So the matrix that is transferred on also matters. One of the projects that we spent a lot of time in our lab working on is called Conserve. This is a USDA NEPA-funded project led by the University of Maryland with very many collaborators from the University of Maryland College Park, the University of Maryland Eastern Shore, the University of Delaware, the University of Arizona. And this is basically a 360-degree look at what's in our water. I can tell you more, but if you go to the website www.conservewaterforfood.org, you can learn a lot more about what we do here. And this is just to show you that what's in our water varies by region and by pathogen. Excuse me. Varies by region and by pathogen. So, sorry, went ahead a couple of slides. So varies by region and by pathogen. I apologize for that. And basically, if we're looking for Shiga toxigenic E. coli, which is E. coli 0157H7, what we have is a really low prevalence across the country from 0.2 to 14%. But if you look at Salmonella and Listeria monocytogenes, you see much higher prevalences based on the location, the geographic region, on the types of water sources that you're sampling in, ponds, rivers, lakes, maybe it's treated recycled wastewater. These are all things that affect the prevalence of these pathogens. And just to take again the Shiga toxigenic E. coli, you can look and see what the geographical difference makes. This is a really large study done in California by my ARS colleagues, Michael Cooley and Lisa Porsky. They had almost 1,400 samples in a watershed in California. They found about 11% of their samples contained Shiga toxigenic E. coli. When we did a similar study region in Maryland, we only found about a 2.4% prevalence of Shiga toxigenic E. coli there. So again, the types of water, the geographic location, what animal, livestock inputs, what the climate is, all of these things can affect the prevalence of these pathogens in the water. We also conducted a large survey here as part of the Conserve project where we looked at between 10 and 35 sampling events over 2 years at different sites ranging from rivers and creeks to ponds and reclaimed surface water. And the main takeaway from this study was that basically we found much higher prevalences of Salmonella and Listeria monocytogenes in rivers than we found in ponds. And this is kind of surprising to me because when we looked at these ponds are really not well kept. They had a lot of moss, a lot of algae. There was many things flowing into it. You would think that just from the visual appearance of the pond, oh, it has to be contaminated with something, but it turns out they're much lower prevalence than say like the rivers that we looked at, maybe because there's less agricultural inputs, maybe the ponds are rain fed, maybe they're there to hold groundwater. I should mention that these are agricultural ponds, and some other authors have found the same thing as well in Virginia and Georgia. So in general, ponds had less pathogens than the rivers, at least the ones we sampled. In another large study that we did, a 4-year study where we looked at survival durations of E. coli in different types of manure-amended soils, what we did was we looked at 3 different research farm sites in the Mid-Atlantic, 2 in Maryland and 1 in Pennsylvania, and this is the map of that region, with different soil types. And basically we tracked 2 different types of E. coli here. We would apply different types of manure to different types of soils, inoculate it with E. coli, and basically look at how long this E. coli could survive in these soils, just to see what matters. Does the manure type matter? If it's poultry litter or horse manure or dairy manure liquid or dairy manure solid, doesn't matter if it's tilled or surface applied. Doesn't matter if it's managed organically. Doesn't matter what year and what site and what season we do this experiment at. We're trying to evaluate all of these things to help support the FSMA rulemaking process about the intervals that you need to have between the application of raw manure or untreated BSAAO and the harvest of crops that are going to be consumed raw. So basically, this was a really big study, 12 separate field trials over 4 years. We got about 6,700 data points, 324 survival profiles. These are what those survival profiles look like after some really nice modeling done by our statistician, Brian Vineyard. And the takeaways from the study were that in general, poultry litter amended soils supported longer survival durations of E. coli. We were looking for the survival time, not the actual levels. So we're looking at the survival durations of E. coli. And poultry litter supported that longer survival duration compared to horse manure or unamended soils. But really what we call the spatial temporal effects, the site, the year, and the season where the study was done were extremely influential in determining that survival duration. And our FDA colleagues in the Decisions and Risk Sciences branch took our same data and looked at it a little bit differently. They were looking at the actual levels of E. coli in our studies. And what their take-home messages were in this paper were that the amendment type, so poultry litter again, predicted longer survival durations, but also longer survival levels. E. coli survived at higher levels for longer periods of time than the other manure sources, dairy and horse manure or unamended soils. But the other thing that was interesting here is that the days of rain since the previous sampling day was also pretty influential. And if you're familiar with the random forest output, this is a variable importance plot here, showing you the different types of factors that went into the model. So in this case, days of rain is the number of days that rainfall actually occurred. It's not actually the total amount. So these are interesting things, and that's really good federal data sharing here. We feel really good to collaborate with the FDA with their ability to do this type of analysis, and they originally supported this work through an interagency agreement, which we really appreciated and allowed us to do such a long-term study. Some other work that we have not published yet, but we just completed and submitted was with, again, a collaboration between the University of Delaware and the FDA and USDA ARS. This is work led by Dr. Pushpinder Litt and Dr. Callie Neal's lab at the University of Delaware, basically looking very similar to our other study where we look at soils amended with poultry litter, composted poultry litter, heat-treated poultry litter pellets, or a chemical fertilizer, getting all of these, apply them to the soil, add our E. coli, similar to what we did in the previous study, and then we plant cucumber plants in that soil. And over 120 days, we take soil samples, and then when the cucumbers are ready to harvest, we take cucumber samples and look for levels of the E. coli to see what happens. And in this case, what we're concerned with is the amount of transfer that we get from the cucumbers, from the soil, which had this E. coli, to the cucumbers. And you can see that we've done this in 2 years, in 2018 and 2019. These are different biological soil amendments of animal origin that we're evaluating here, but that's less important than what these black lines are right here. And this is box and whiskers plot. The black line is essentially the median value, 50% above, 50% below. And you can see here that in 2018, our median values of all their different treatments are much higher than they are in 2019. So again, this goes back to the role of year and season on the survival of pathogens in the pre-harvest environment. And this differs a lot by year. You can see that we're getting some decent transfer here of the E. coli that we know that we put in the soil, and it is transferring to these cucumbers. Still a pretty low level, 2 to 3 logs CFU per cucumber. But in 2019, we just have a couple of outliers that get that high. The median values are basically just at 0, right above 0. So what accounts for this difference in year? Well, if you look at the total amount of rainfall and the rainfall patterns, both of these things I think are important. I think the total rainfall during season may affect transfer. And if you look at here in 2018, we got about double the amount of rainfall that we did in 2019. But it may not just be that rainfall amount. It may actually be the pattern that this rainfall is organized in. So you can see here in 2018, you can see that we don't get a lot of rainfall. This is rainfall measured over 10 days, the cumulative amount of rainfall every 10 days. And you can see there's not a lot of rainfall in the first 30 days. But then what happens is we go up here between days 31 and 40, and we get really large rainfall days here between days 31 and 40, all the way up to 61 and 70. And almost the opposite happens in 2019, where you get more rainfall at the beginning, but then those rainfall amounts tend to fall through the study. And this, again, may affect the survival dynamics of the pathogens in soils. And what we'd like to say is that it's a combination of all of these things. It's the rainfall, it's the soil, it's the location, and it's also the type of biological soil amendment that you're putting into the soil that's affecting the survival of these things. So it's a very dynamic environment. And just to conclude, this is basically what we say. The spatial temporal effects can affect how long pathogens survive in soil. They can be transferred in water and then eventually transferred to a growing crop. And these are dependent on spatial temporal factors, but also specific agricultural effects, like the soil type, like the manure type, like the crop being grown, whether it's grown on the floor, whether it's high or low to the ground. And really what we're looking for are agriculturally predictive variables that can predict the survival of E. coli, Salmonella, Listeria in these matrices. Is there something, a nutrient level, the rainfall amount, a specific chemical indicator, or a non-microbiological indicator that a grower could actually use to say, hey, my crop might be more at risk of contamination this season compared to others, or when I see this level go high, I need to pay more attention to the microbial contamination issues that may be occurring. And we're hoping that some of these large studies that we're doing can inform on those types of variables. But we really have to do these large studies first, go back and retrospectively apply them, and then do smaller studies evaluating those specific factors to see if this actually works out. So with that, I'd like to thank all the funding sources that have been instrumental in doing all of in doing all this work. Our ARS base funded project, the Conserve project funded through NIFA, the interagency agreements funded by the FDA, and also our partner institutions, the University of Maryland Eastern Shore, the University of Delaware, and we have many collaborators at many different institutions across the country. North Dakota State, University of Maryland Eastern Shore, University of Delaware, University of Maryland College Park, and also our great staff here at USDA ARS. And with that, I'll conclude my talk. I just want to thank you all for your attention, and I'm happy to answer questions in the roundtable.
Okay, some really interesting data there. I have at least one question, but I'll leave opportunity for folks that are on today to put their questions into the chat box first. So please do enter those into the chat and we'll try to get those answered. I did note that Manon is on this morning, so he's available to answer those questions live. Well, I'm not seeing anything come in just yet, Manon, but so I was intrigued by the data analysis where he showed days since rain and how that was related to, I'm going to say bacterial counts. Hopefully I'm not misspeaking on that. But so can you share a little bit more what that relationship was? So was it days since rain that you're more likely to see the bacteria or less likely?
Yeah, no, Joe, that's a really good question, and thanks for it. So it's it was the number of days that rainfall was observed without regard to the amount of rainfall, right? Just the number of days, whether it rained or not, between the time that we took one sample, the time that we took the last sample and the next sample. And that number of days basically influenced the count. So the more number of days you had, the greater chance that you had higher bacterial counts. And the less number of days of rain that you had, the greater chance that you had lower numbers of bacterial counts. So more days of rainfall, higher populations, essentially. Less days of rainfall, lower populations. That's what that variable means. And that's not related to the amount of rainfall that was observed in that interval. So, and then a question just came in, and that was in soil, not the water, correct? Yes, that's correct. That was in the soil containing different types of biological soil amendments.
So the potential practical explanation on it would be that more rainfall, more potential splatter up onto the plants, which would then cause the contamination.
Right. So two different studies. One study where we're just evaluating the persistence of E. coli in soils containing whatever amendment they have, right? So in that case, the rainfall is increasing, like keeping those E. coli levels higher. And then the second study where we're looking at the actual transfer of E. coli to the cucumbers, yes, it seems like there's a relationship there between the amount and the pattern of rainfall and the transfer to the cucumbers. Then the more rainfall you get, the greater likelihood you get transfer. However, I think the one thing that I didn't do a good job pointing out in that second cucumber study is that I do think that there is a very dependent effect of the specific type of soil amendment. And that, so it's an interaction between the rainfall, what's in the soil, and the transfer to the fruit.
Sure. Okay. And we did have one more question come in. Says, is there any way that we can ensure that E. coli will not transfer to a crop such as lettuce when using amendments?
So Ensure is doing a lot of work in that question. I appreciate the question because it's a really good question. I think ensuring is, I don't think you can assure. I think you can minimize the risk by using some of the intervals that are recommended by the National Organic Program, the 90 or 120 day interval that hopefully Dr. Ingram talked about in his talk. So that would be one thing. That's the direct route, from contamination in the actual manure, essentially, going to the soil. But I think that we're seeing that it's more complicated than that there is this climate interaction. And there also is this whole issue of adjacent land use, which I think one of the speakers later on this morning will address as well. So, but I think following the guidelines is a good way to minimize that. So basically applying your manure as early as you can and still get that nutrient value that you need to your crop.
Great. I think that's the last of the questions that came in. Great talk, and try to keep us on time. I think we're going to go ahead and move forward. Our next speaker is Laura Strawn, and she's going to be talking about, Laurel Dunn. She's going to be talking about salmonella and poultry litter and food safety.
Hi, I'm Laurel Dunn. I'm an assistant professor and extension specialist focusing on produce safety from the University of Georgia in Athens. I'm actually going to discuss a project that was done in Michelle Daniluk and Keith Schneider's labs at the University of Florida, assessing the prevalence and concentration of Salmonella enterica in poultry litter collected from farms throughout the southern United States. I am going to touch on Georgia poultry production, though, because I am in Georgia and because poultry production is a big deal in Georgia. In 2018, Georgia was #1 in the country for broilers and for hatching egg production. We were #4 for pullet production, and we were #5 for table egg production. So overall, this resulted in a 2018 poultry and egg farm gate value for the state of Georgia about $5.7 billion. So a lot of chickens, a lot of eggs are produced in Georgia. And with that, a lot of waste material. And the primary waste material in poultry production is poultry litter. And so this is comprised predominantly of bedding, which depending on the region you're in, can be sawdust, it can be waste products from peanut production, so like peanut hulls, rice husks, things like that. It also includes dropped feed, manure from the birds that are living on the litter, and also mortalities when birds die and either die in the house or are taken out and added to a litter pile. Poultry litter is a very high nitrogen source. And so when it is sold as fertilizer or as a soil amendment, at least in Georgia, the price ranges from about $15 to $20 a ton. In the United States, there are 10.2 million tons produced annually. 2 million tons are produced in Georgia every year. So we have a lot of poultry litter produced in the US and particularly in Georgia. The safety of poultry litter is being scrutinized predominantly because it is often applied to crops. And while sometimes members of the produce industry want to use it, there are concerns about it containing salmonella or other foodborne pathogens that might be conveyed on fresh produce to the consumers. We're particularly concerned about some of these pathogens because once they are introduced into the soil, if poultry litter is introduced to the soil and it contains salmonella, these pathogens can persist for long periods of time, weeks to months, and eventually go on to contaminate produce. And so this can happen when litter is directly or land applied. It can also happen indirectly if you get runoff from rain events, or if poultry farms tend to be in the same watershed as produce farms, you can get contamination of water sources or spread to produce through those avenues. And so while we were assessing the safety of poultry litter and biological soil amendments of animal origin, or BSAOs, we had a few questions that this project was really gearing up to address. And so one was, how frequently does Salmonella enterica contaminate poultry litter? At what level does this contamination occur, which can be useful for calculating things like die-off rates or determining risk? How long can Salmonella persist in litter? What serotypes are commonly present? Because different serotypes have different characteristics or different affinities for different produce types in some cases. Does contamination fluctuate according to season? And do regional differences account for Salmonella contamination? And so going into our methodology, we were particularly interested in the Southeast, because at the time I was located in Florida with Michelle and Keith. However, we kind of had to branch out to where the farms were. So we started with Florida, Georgia, and Alabama. And then we ended up recruiting a couple of farms from Texas. And so we did this by cold calling some large poultry companies and composters. We used our extension resources. We found contacts at a nationwide conference. And we really kind of hit the jackpot when we teamed up with the State Poultry Association. That's how we had so much success in Southeast Alabama, where we were able to recruit 6 farms. So we ended up with 13 farms in total. And so we had kind of 2 separate sampling methods based on what the situation was at each farm. So a lot of farms would pile their litter in these storehouses or out in the open. And so we would record, whether or not these piles were protected, the depth from which samples were collected, temperature, the facility types, so do they have breeders or broilers, and then just any other storage conditions, weather conditions. And so we would identify 3 piles at each farm. And so from each pile, we would collect 7 samples. 2 of those were toe samples. Their poultry litter. So if we had to go into the actual house to collect it, we just kind of divided the house into thirds. So we'd collect our first 7 samples from the front, the middle 7 from the middle of the house, and then our final 7 samples we'd collect from the back of the house, just collecting samples from the floor, or in instances where we dealt with poultry or pasture flocks, then we would just collect in kind of the same quadrants, but around the pasture itself. So I won't go into our methods, our lab methods too much, because we don't have a lot of time, but we took 30-gram samples of litter, and we initially screened them for presence of or absence of Salmonella. So we did this by eventually streaking on XL24 and then taking any presumptive positive isolates and doing PCR confirmation. And if we got a confirmed positive, then we would begin with our NPN, or most probable number calculation, again using 30 grams of litter. and screening again onto XLT4 within a PCR confirmation. When we kind of wrapped up the experiment, we sent the isolates off for serotyping. So for our kind of total salmonella prevalence overall, we ended up with 490 30-gram, I guess, litter samples collected from 70 piles or surfaces from 13 farms. And so what we found was overall, 6 of the 13 farms, or 46%, were positive for Salmonella enterica. This came out to be about 7% of samples total, so about 33 samples of the 490 ended up being positive for Salmonella enterica. We found that out of the 13 farms, Alabama had 2 out of 7 that were positive for Salmonella. 1 farm out of Florida was positive. Both farms out of Georgia were positive for Salmonella, and 1 of those farms was the only farm that ended up being positive on both visits. So all the other farms that were sampled were only positive on 1 visit if they were positive. And then 1 Texas farm yielded Salmonella. We found that we collected Salmonella more frequently out of samples that were collected in the fall and winter months, so 12.3% or 32 out of 259 samples that were collected in the fall and winter were positive for Salmonella, compared to roughly about 1% positivity from spring and summer collected samples, or only 2 samples out of 231 were positive for Salmonella in the spring or summer months. We ran a logistic regression and found that Salmonella was 96% less likely to be found in toe samples, or 95% less likely to be found in gut samples than it was to be found either from the floor of a poultry house or from the ground from a pasture flock. So we were a lot more likely to find Salmonella from our pasture flock, ground, or from the actual floor, and not from piled litter. And so we kind of have the numbers here, and nice little easy to read pie charts where we have 33% prevalence on farms that had piled litter. So 2 out of 6 farms were positive for Salmonella when they piled their litter, compared to 4 out of 7 farms that were positive if the litter was just collected from the poultry house or from the pasture itself. And we also found that, again, piled litter, we had about 3% of samples positive. So 6 samples out of 231 were positive in samples that were piled, compared to closer to about 10% of positive samples when they were collected directly from the floor or from the pasture. So we also found that Salmonella was not detected in litter that was older than 21 days. And we found that for every 1-day increase, the likelihood of finding Salmonella in those samples decreased by 5%. And so some of this is likely some of the reason why we found Salmonella more frequently when collected from the floor or from the pasture, because usually those samples were younger litter samples. Birds had been more recently on those samples or on that litter. So some of the houses either had birds at the time of collection, or if they hadn't, birds had recently been in there and had been moved out, and either the litter was waiting for the next flock or was waiting to be removed, and, waiting on equipment or workers to remove it. We also found that, a lot of times when we were digging in the litter, especially for the gut samples in those piles, A lot of times the temperatures range from 130 to 140 degrees. This was particularly evident in the winter when you dig a hole and steam would start coming out from the interior of the pile. And so there was some natural composting going on, even though these weren't validated composting procedures going on. Just by the nature of this litter being stacked, it was already starting to undergo that biological decomposition process that was generating heat and likely killing some of those pathogens. We also found that our concentration of salmonella fluctuated greatly among samples within the same farm or even within the same pile. And so we found that we got a range from less than 1 to greater than 280,000 most probable number salmonella per gram. And these again could be, one sample could be the less than 1 sample could be the greater than 280,000, all within the same pile or all within the same farm. So we got great fluctuation in the concentrations that we saw. We also had several serotypes that were identified. And so from Alabama, we had brain drop in Kentucky. Florida, we tended to just see St. Paul. Our Georgia farms, we found Salmonella enterica serotype Kentucky, Michigan, and Septemberg. And then from Texas, we had 4 different serotypes, including Mbandaka, Kiambu, Anatum, and Newport. So in conclusion, overall, we detected salmonella in about 7% of our samples. Our salmonella concentrations were highly variable within our farms. We less frequently saw salmonella from farms where the litter was piled. So again, only 33% of farms that piled their litter were positive for salmonella compared to 57% that didn't pile that were positive for salmonella. And we identified 9 different serotypes across the 4 states and 13 farms. And so just kind of a few additional thoughts before I wrap up. For one thing, and this is probably geared towards extension or people that really work in food safety produce industry, there is a disparity in the definitions of compost between produce and poultry producers. And we found that consistently while going to collect samples. A poultry producer would not hesitate to offer to take us back to the compost pile when really they just meant the dry stack. And produce growers, on the other hand, when they say compost, they're looking for that FDA-defined, scientifically validated process. And some other things. I mean, there are many other factors that kind of confound this and that need to be looked into further. So our poultry litter composition, either the material can make a difference, but we saw differences between our breeder farms and our broiler farms. So that is going to also impact some of the prevalence of some of our pathogens in litter. So if you have any more questions, you can contact me. Like I said, I'm at the University of Georgia. Here's my email. Thank you for listening.
Okay, we'll go ahead and take some questions now. Again, go ahead and put those into the chat box. So I guess I'll ask one while we're waiting. So I think it was some information in Mannon's presentation. Is it related to poultry litter and being maybe was more likely that you would observe certain pathogens on either the soil or the crop? And yet Laurel's talk said that, it seemed like there was a pretty good kill in or die off in the first few days. So can you guys connect those dots for me, or am I inappropriately connecting those dots?
So you're asking, where mine says it seems like they die off pretty quickly, and Manon's seems like they linger a little longer. Is that kind of what you're asking?
Yeah.
I mean, we do see those differences, and a lot of what we saw with the stuff I was looking at dying off was it was in big piles for the most part. So you're getting a lot of that composting activity. There's a lot of heat in those piles. Whereas I think with Manon's work, a lot of it was done in field where it's spread out. You're not getting that heat from the natural biological composting thermophilic process that's going on. So we're kind of comparing two separate things there. We would expect, I would think, and it's typically what we see, if you're field applying this, you're just not getting that heat, and that's, I think, why we're seeing differences in this. And this is why it's important to have that validated process when it comes to composting, because you never know what you're going to get really, as far as what will die off, where things are in the pile, and so forth.
Great. Okay, thanks for helping me with that. I haven't seen any other questions come in. Question was, weren't the poultry piles also contained under cover?
Not always. For the most part, they were. And so that was another thing we tried to record. And our statistical analysis found that while whether or not a pile was covered or exposed to the environment, it didn't significantly impact whether or not we found things. But it was still important to the statistical model. So we still recorded that. A lot of our piles were undercover, where, yeah, we were kind of comparing a few pastures, which were obviously exposed to the environment. So, yeah.
Okay.
Laurel, if I could add something, we did the sister study up in Gainesville. This is Keith Schneider. The poultry litter behaves really different than the bovine, because we were doing bovine and chicken at the same time. We had another study going, plus the one that we were collaborating with, and there is a huge difference between the two sources, and they do behave radically different.
And the difference too is with the bovine, we're looking at pretty much straight feces, whereas with the poultry litter, you're looking at predominantly solid shavings or peanut material. I think for our study, it was mostly the sawdust that we saw. That's then mixed in with the manure. So very different information you're comparing.
Okay. Great. Okay, thanks. Thanks for being on with us this morning. Okay, we're going to go ahead and move on to our next talk, and it's by Keith Schneider. It's going to talk about post-harvest use of sanitizers.
Hi, this is Keith Schneider. I'm a professor in the Department of Food Science and Human Nutrition at the University of Florida. I'm going to give a talk on post-harvest water quality and disinfection through disinfectant use. One of the reasons we spend so much time with post-harvest water is just its ability to make a very small problem into a larger problem. Communal water baths like the flume that you see pictured, or ice used to top produce coming in from the field to remove field heat, all have the ability to spread contamination that might be isolated to a small amount of produce and spread around to a larger amount, thus increase the potential for having a foodborne outbreak. Water is often a common source for contamination in packing sheds and along the field and packing sheds. Ice is frequently overlooked. Again, it's typically made and then stored on the concrete, right on the ground, and then scooped up with a front loader. So we have to look at that as a potential source of contamination. And any water reuse, flumes and brush beds, anytime we recirculate water, we have to be very careful that we handle that water properly. Again, don't use those as sources of cross-contamination. So let's look at some numbers here. We look at some of the multi-state outbreaks, and this is some of the more recent information that was gathered in 2019. This goes from 2010 to 2017. You see the largest number of multi-state outbreaks is associated with sprouts, and then followed by some of the usual suspects: melons, lettuce, high in cucumber, and then leafy greens and tomatoes. So if we look at this chart, the distribution is, again, fairly consistent with what we've seen. But if let's take sprouts up for a moment, we just look at row crops. Greens, when we consolidated all those, become one of the largest offenders, with about 37% of all the multi-state outbreaks associated with greens. And we just have to go back to the 2018-2019 romaine lettuce outbreaks to kind of get a sense of that. Then melon and papaya, cucumbers and tomato. So again, these are some of the usual suspects. And one of the things we want to do, especially with post-harvest water, is to limit the potential for a foodborne outbreak. Now, it's almost impossible to prevent a foodborne outbreak in the field. While we do have produce safety rule and programs like the Leafy Greens Marketing Agreement and the PGAP program in Florida, getting or preventing 100% is almost impossible. So the goal of post-harvest water is to limit those or limit the potential of those as much as possible. So these are straight out of the PSA training guide. So just wanted to point out some of the rules, what's in the rule. For post-harvest water, you can have no detectable generic E. coli in 100 mils. The Food Safety Modernization Act, Produce Safety, does not require water to be treated. So any antimicrobial products are considered good practice, and they're also considered to be very valuable in that they can prevent the buildup of microorganisms, aka biofilms, on food contact surfaces. The one thing I want to point out on this slide, though, is that on single pass water, it's not required to have any type of sanitizer at all. So even though it's recommended, it is not required by law to actually use a sanitizer. Now, that's not the case for the PGAP program, where you do have to use a required amount of sanitizer in the water. If in fact you do use any type of wash process. Same goes for recirculated or batch water. Again, the water quality, straight out of the produce safety rule, is no generic E. coli per 100 mils of sample. But you have to maintain that water quality throughout its use. So if I'm recirculating a bloom tank for 6 to 8 hours, even though at the beginning of that processing day I had 0 E. coli, even though I'm recirculating it, I have to maintain 0 E. coli. Interestingly enough, that treatment is not required to maintain water quality or to prevent cross-contamination. And the question is, well, how do you do that? And the answer really is you don't. But again, if you are going to use an antimicrobial to meet this water quality standard, you must be labeled for the appropriate use. So it has to have that EPA registration number, and it has to be labeled for the washing of fruits and vegetables or the treatment of clean water. But it has to be for specific use. If you use batch water or circulating water, you have to have some way of changing it or some frequency of changing it. And again, the use of these sanitizer antimicrobial products are really recommended for the prevention or buildup of biofilms on these surfaces, which again can exacerbate the microbial load and potential foodborne outbreaks. So things to consider. Sometimes you hear you want to start with water equivalent to drinking water. That's not necessarily true. The water used for post-harvest does not have to be potable. It just has to meet the 0 E. coli standard. So sometimes you will hear drinking water quality. That's not the case. All we're looking for is no generic E. coli per 100 mils. But that does then exclude the use of untreated surface water. But we still don't have to meet the municipality regs, which are much stricter than what the PSA outlines. If you're going to use a sanitizer, we have to make sure you're using a food grade sanitizer. Again, we mentioned EPA registered for intended use. And while it's not required, it is recommended. If we're going to use water in a flume, hydro cooler, or some type of chiller, we have to have some frequency that we're going to exchange that water when we think it's no longer fit for use. There is also the requirement or recommendation that the temperature is acceptable to reduce any type of infiltration due to the temperature differential between the recirculating water and the fruit itself. And then lastly, the sanitization of any food contact surface. And again, post-harvest water goes from tools, scissors, or shears used in the field, all the way to the equipment that's used for packing. And there's just a couple of links for you to look at. To get my water to the level I would like, I could use some type of antimicrobial device. I could use UV light, an ozone generator, some type of filtration system. I can use EPA registered antimicrobial pesticides or sanitizer solutions. They can be chlorine based, seems like sodium or calcium hydrochlorides. I can use peroxyacetic acid, hydroperoxide, again, ozone. So there are any number of choices out there that you can use. You just have to pick one that is safe to use and is allowed to be used on the crops that you're harvesting. So what I want to do now is I'm going to specifically talk about chlorine-based sanitizers because they're the most frequently used. And what we typically are looking for is a residual in the water. And there's a couple of different things we want to consider. One is how we're going to measure that and use things like test strips that you see in the picture there. I can use a handheld spectrophotometer, that picture on the upper right. But again, we just want to be doubly sure that it is a food grade chemical, that you're using it at the appropriate levels, putting the manufacturer's recommendations and what it is registered at. And anytime we use any type of antimicrobial in the water, we want to make sure that we have some type of monitoring in place. Again, if we're going to use a particular chemical, we want to make sure that it's applied to the standard that we've adopted and that it's being applied at the rate that we know is efficacious. One thing to consider anytime you're using any type of sanitizer in a recirculating system is that there's going to be organic load, and that organic load is going to compete for the chlorine or the free chlorine or the oxidant in the water. So this is a concept known as breakpoint chlorination, and it's this is typically something you think about again in wastewater treatment, but it applies to any type of chlorination like you would use in a flume tank. As you begin to add chlorine to any system, the first thing it's going to do, that chlorine is going to start to bind up and oxidize materials in the water. And you're going to start producing combined chlorine compounds. And if there's nitrogen in the water, chloramines, trichloramines. And at a certain point, once all of the available material has been oxidized or bound up, then and only then do we get free chlorine being released. And that's known as the break point. You can kind of see that on that graph there. And as you add chlorine after that, we see a rise in the amount of free chlorine that we're able to read within the tank that we're using. So whenever we're adding, we have to kind of consider that there's going to be a certain amount of chemistry that we use that's not going to be being used for disinfection at all, but rather just going to go to oxidizing those organic compounds that are in the water. And as the quality of the water changes throughout the day, we have to take into account that we're going to have to add more chemistry per hour to overcome the organic load as that water gets dirtier and dirtier. At a certain point, it's going to be to a point where the chemistry is not going to be efficacious at all. And this is why when we get to a system, we want to make sure that we change the water on some regular basis. So if we look at total chlorine, we put in a certain amount, let's just say 100 ppm. But that 100 ppm, especially if we're putting it into a water that's not quite clean, we're going to get a certain amount of free chlorine, and then we're going to get the combined chlorine. So who knows, maybe out of that 100 that I put in, I may get 60 free chlorine. So you're thinking, okay, I'm putting 100, I'm going to get 60, that's what I'm going to work with. But there's also another problem that you have to consider. Hypochlorite, things like sodium and calcium hypochlorite, will produce hypochlorous acid, that HOCl compound, and that's considered free chlorine. But there's also a portion of that that is in the ionic form, the OCl minus hypochlorite ion, which is also considered free. The difference is that HOCl is much more efficacious than OCl minus. So out of that 60, depending on my pH, I may only have half of that 60. So now out of the original 100, I may only have 30 ppm of HOCl, which is my free and active form of chlorine, actually doing any type of disinfection. So here's what that looks like. Anytime I put any type of hypochlorite in the water, I'm going to produce hypochlorous acid. That's the species I want. And whether it's sodium, which is in a liquid form, calcium hypochlorite, which is in tablet form, or even chlorine gas. When I put any of those into water, what I'm going to end up with is HOCl. That HOCl is going to differentiate or dissociate into OCl minus. And this really depends on the pH, and that the curve that kind of shows that is here, where around pH 7.5, 7.6, I'm going to get the 50-50 point, where if my pH is at 7.5, 50% is going to be in that HOCl form and 50% is going to be in the OCl minus form. So whenever you're trying to figure out effective dose, you have to first figure out, again, how much I need to achieve breakpoint, and then whatever my free chlorine reads, I can tell how much of the active form is by just knowing the pH and using a chart like this. So if I want to be more efficacious, I can just I can lower the pH. If I lower the pH to say 6.5, I'm going to have roughly 90% of the active form, which again increases the efficacy, but I have to add an acidulant. If I let the pH go up, say to 8.5, the reverse happens. I only have about 10% in the active form and 90% in the inactive or less active form of hypochlorous acid. Now, what if you say, I don't want to mess around with pH at all? Well, here's an example where more is not better. So let's start out where we have 200 ppm. That's our total chlorine that we're going to put in the system. We take a reading and we're at pH 6.5. Let's just say this is all in the free form. And then I'm going to use in another tank 2000 ppm. But since I'm not adding any acid to this system, I'm just going to let the pH naturally settle. And at 2000 ppm, the pH would probably be slightly higher than 8.5. It might be 9.5. But for the sake of this argument, let's just say it goes up to 8.5. So in example one, I will have 90% of that 200 ppm in the active form, or roughly about 180 ppm in the HOCl minus form. In the second example, I'm going to end up with about 10% in the active and free form, which is roughly 200 ppm. So I've added 10 times the chemistry for about the same efficacy. Almost the same amount of active, free, and active chlorine are left over. So if you visit this, by just adding an acidulant to that initial 200 ppm solution, I can increase the efficacy up to 90%, and I save a lot of chemistry. Now, what we don't want to do is continue to add acidulant. Well, 6.5 is great. Why don't we go to 5.5 and make it 99%? Well, as we get closer to 5, that curve starts to drop off and the amount of HOCl begins to drop again. We start producing chlorine gas. And at that point, we end up with a very dangerous situation one of the reasons when we talk about household disinfection, we tell people never to mix vinegar and bleach because you can get the elution of chlorine gas, which can be a major health hazard. So our target zone typically is between 6 and 7.5, but most applications 6.5 to 7.5 usually gives us anywhere from that 80 to 50% in the free and active form is what we're looking for. Now, one thing we have to be very cognizant of is the amount of organic material in the water, and that will quickly build up over time. And the more organic material we have in the water, the less of the sanitizer we're going to have to react and kill the microbes. So we want to keep an eye on how much we have in there. And one method people commonly use is ORP meters, oxidative reduction potential meters. And as the ORP drops, this can be programmed into a pump to turn on and apply more chemistry and to bring the ORP back up again. And that's a good way of distributing some chemistry. ORP doesn't give a great estimation of what amount is there, especially as the organic matter builds up. That ORP measurement becomes less reliable, but it's still a good way of introducing sanitizers into a system. But you got to be very cognizant, and then we have to develop some type of frequency. And again, we could use things like a handheld turbidity meter, or even just a simple comparison chart to determine the frequency which we want to change our wash water with. As with any process, again, we want to monitor the sanitizer use, make sure that we take routine measurements. Even if we have an automated system that's using something like ORP, we still want to take maybe hourly measurements of that system to make sure we're getting an accurate reading of the sanitizer, especially in a recirculating system, and that we're getting the efficacy that we're looking for. Anytime we're changing the water, water quality testing, especially again if we're using municipal water and even well water, to fill these tanks again to make sure that we meet that minimum quality of no E. coli per 100 mil. So in summary, one, post-harvest water should start out clean. Again, we're looking for the level to be 0 E. coli per 100 mil, or no detectable E. coli per 100 mil. In a single pass or even recirculating system, we have to maintain that water quality. And remember, it is not required by FDA that you use a sanitizer, but it's highly recommended. And again, it's almost impossible within a recirculating system to not use some type of either device or sanitizing agent to maintain that water quality. Don't forget about ice. It's one of those things that are often forgotten about. Those systems need to be clean. We have to make sure that, again, if we're topping produce coming out of the field, then again, we're making sure that ice is clean. Preferably, we're not contaminating it by producing it and storing it on the ground. It's a very difficult situation to kind of manage. And lastly, just make sure they're using EPA registered chemistries and using them correctly. So that's my time. Again, I'll be able to answer questions if we have that capability. And thank you very much for your attention.
Okay, thanks, Keith. Questions for Keith with regard to sanitation?
What is the cheapest method for monitoring chlorine levels? One of the cheapest that I've seen, and the one I see most often, is using chlorine strips. Again, if you're going to use chlorine strips, you're going to have to make sure that they're registering for free chlorine, not total chlorine. The bad news about using chlorine strips is they are sort of frowned upon since they don't give you a number, they just give you a range. I think United Fresh in their latest tomato metrics document recommended not using strips, but those in fact are the cheapest. Other than that, you're going to have to get some type of colorimetric test, similar to, again, sort of like a pool testing kit. The one I use is about $1,000, but again, even the cheaper ones are still kind of expensive, $200–300. And then you also have to do a dilution. So as far as the cheapest method, strips, but again, they're not the most favored. So that's how I'd enter that one. Do you see chlorine falling out of favor as a pesticide, because of the issues with pH and free chlorine. I really don't at this point. One of the biggest things, if it's going to fall out of favor, is like, again, the increase in organic production is you'd have to you can still use chlorine, believe it or not, in organic production. You just have to use a very low amount, and that's really not efficacious to prevent cross-contamination within a flume system. There are concerns about disinfection byproducts, halogenated amines, trihalomethanes. You've probably heard about those. So from an environmental standpoint and from maybe a consumer demand, maybe you see a reduction, but the fact that there's very little evidence that there's been any direct health implications. And in fact, it's cheap and effective. I don't see it being replaced anytime soon on that one. What chemistries are less affected by changes in pH? I just mentioned the one, peroxyacetic acid, PAA products. They are more tolerant of pH. In some cases, they're more tolerant to organic load, but my student literally just finished a study and submitted it yesterday. The PAAs are affected by organic loading, as are free chlorine, but just not to that extent. And the upside of the PAA products is, in fact, they are a little bit more tolerant of organic load, and in fact, they can be considered organic. So the couple of benefits there for that question. But yeah, things like PAA are less sensitive, things like ozone, but ozone's kind of a pain in the patootie. I wouldn't recommend that unless you have a high value product, because that's really hard to manage and you have worker health issues with off-gassing. Any other questions? And again, firstly, I want to thank everybody, because again, I've been joking with some of the friends who are on this, that I've stolen so many slides from people. I don't know who. So I want to thank everybody who I have borrowed some slides from. I greatly appreciate their help in all this.
Okay, thanks, Keith. Well, I certainly learned a lot, particularly about all the things that affect the efficacy of it. So lots to adjust to in the food safety arena. So
one of the things we don't think about as much, and we really should, is breakpoint chlorination. If you haven't heard about it, read up on it. It really kind of gives you a good indication of how, when you're dosing a system, how that chlorine is actually being used up prior to you actually getting good efficacy in your wash water.
yeah, great. Okay, thanks. Okay, we're going to move on to the last talk this morning before we go into our bio break, and it'll be by Laura Strawn. It'll be factors affecting prediction of pathogen hotspots. So go ahead and boot that up.
Good afternoon. My name is Dr. Laura Strawn, and I'm an associate professor and extension specialist at Virginia Tech. And I am very happy to be here with you all virtually at this year's SARA Conference 2020 to present my work, Factors Affecting Prediction of Pathogen Hotspots. We'll start with a little bit of an overview of GIS, or Geographic Information Systems, make sure we're on the same page. And then we're going to talk about two particular studies where we've started to use this in produce food safety to predict pathogens. And we'll focus in on Listeria monotypogenes. And then we'll end sort of with our final thoughts and where we see this going. Geographic Information Systems is a computer system that is used for storing, capturing, checking, and displaying data related to positions on Earth's surface. GIS can also show many different types of data on one map. And this enables us to be able to easily see, analyze, and understand patterns and relationships. And when we're thinking about food safety, and specifically in pathogens in an environment, it's all about seeing those relationships and those patterns to predict where are those pathogens going to be. Since our working hypothesis is that pathogen prevalence is not uniform in the environment, we know that there are pathogens in the environment, but we figure that there are certain times of year and certain locations where they'll be more prevalent than others. And once we can identify those, that'll help us with food safety mitigation. So with GIS, it supports various data, both remotely sensed from satellites and radar. A lot of it is publicly available. We can input census data, surveys, we can analyze multiple data sets and develop algorithms that we can use for predictions. And so the graphic to the right shows you street data, building data, vegetation data, and it combines those all into an integrated visual where you can actually be able to look at complex interactions and sort of this 3D view of the world. I think it's worth noting, one of the first applications of GIS was actually used in public health. John Snow, we're going to go all the way back to the London-based physician, 1854. They were having a large cholera outbreak, and he was able to take and demonstrate spatial associations between cholera cases and a single water supply. From there, once they identified the single water supply, they were able then to eliminate access to this water supply, ceasing the outbreak. And so back in John Snow's day, they took cholera deaths. These are the red dots on one of the graphs. All of the blue dots are the pumps. And you were able pretty quickly visually to see that all of the cholera cases were surrounding the Broad Street pump. In today's world, we can use contour plots and mapping software to visualize these spatial associations. We have an intensity plot, and you can easily see that the Broad Street pump is where all of the cases are clustering, or the highest intensity in red. From there, eliminate the Broad Street pump for your water, and outbreak solved. And so this is sort of how we can utilize some of these spatial associations to be able to predict what's causing a problem, in this case, the source of the outbreak, and how we can refine our mitigations. So let's look at these in two examples in produce food safety. One, our first study, we wanted to look at the prevalence, persistence, and diversity of foodborne pathogens in the produce pre-harvest environment. And then we wanted to start and utilize some of these prediction model builders to identify factors, whether they be landscape features, soil characteristics, or meteorological events, and really predict a higher prevalence of where we would see this pathogen. We're going to focus in on Listeria monocytogenes today. It's a perfect one to use. We can find it. We know it's in the environment. So now our goal is it uniformly spread across the environment, or can we actually use predictions based on these factors to pinpoint where it'll be higher or lower? Using this, we took a combination of field studies and computers. We did a large environmental sampling effort. You can see pictures where we took the date of when we sampled, the location, and we took drag swabs, which are basically a good capture of the topsoil. We took soil samples to get underneath what that microclimate is like. We took water samples. We were able to pull remotely sensed data, so you can pull a host of weather data with the date, with that location. You can pull landscape features, how close or far away are you from certain things, and then also we can pull characteristics on the soil. And then we can do some statistical modeling to kind of figure out what's going on, right? How can we use this for prediction? So one of the first things that we did was we built these classification trees based on high and low Listeria monocytogenes prevalence. So overall in the whole study, we found 15% prevalence of Listeria monocytogenes. Again, this is not to be unexpected. We know we can find Listeria in the environment, especially if we go out looking for it. What we wanted to know is, could we actually figure out areas where it was higher or lower based on certain factors? So through our computer modeling, we were able to generate a list of factors that were able to parse out high and low prevalence locations or times of year. So our first thing where it divided basically a high prevalence or a low prevalence was based on your location to water. If you were close to water, you had about a 40% chance, 39% chance prevalence of Listeria monocytogenes, versus if you were far away from water, that predicted prevalence dropped down to about 12%. You can also see that we started to make associations based on weather. So if it was cooler, you were able to find more Listeria monocytogenes. And then one of the interesting things is it started to break it out based on proximity to certain landscape features like urban proximity, whether or not you were around more cities or impervious cover, right? Roads, concrete structures. We were able to see that you could have, if you were close to those types of urban factors or features, you had a predicted prevalence of 20% versus if you were farther away, only 10 meters, 5%. So not very far, right? 10 meters is 30 feet. So you have to be pretty close, right? So where you see some of these fields butt up right to urban features. We are also able to see soil, available water storage and soil. So we started to get our first soil characteristic where we saw if it was a higher available water storage, we were able to get 30% predicted prevalence versus 10%. And then we were able to see the pasture proximity. If you were close to pastures, about 50%. And then if you were far away from pastures, 7.5%. And so we really wanted to be able to validate these models and figure out, would this work, right? We did this research study, we came out with our factors. Let's try to apply this to commercial ag. So we enrolled some commercial growers who were kind enough to let us take their farm fields. They were able to give us maps of their fields and tell us where they owned land. And we were able to build these predictive risk maps based on the different features that we had identified in our models, which was close to water, close to developed areas, low or high available soil moisture, and close to pastures. And what you can see, is that our predictive risk map shows that there's going to be different risk levels, higher predicted prevalence areas versus lower, which is what we hope to see, that you don't have this uniform risk. And so we wanted to go out and then validate it. Well, we went out and took samples in all of the higher areas versus the lower areas to see if we could, in fact, validate these models. And we were able to show that, yes, areas that we thought, for example, this little blue area of this particular farm, not a very large area, but here we were able to find significantly more Listeria monocytogenes than, say, other areas that were predicted to have a much lower prevalence of Listeria monocytogenes. So this was really exciting for us to see that at least in the Northeast, where we had some of this data, we were able to start to get these predictions that were showing up correct. This also allows us, where farmers can then decide and implement different mitigations, right? Based on, whether or not we can do different, plant different crops there, whether we can do different rotations. Is there a way where we can try and do better laser leveling of fields to reduce some of that pooling of water or whatnot. Various different mitigations, and all of this is really tailored specifically to what you see. So our second study, we really wanted to kind of hone in on some of these weather and landscape features. We wanted to continue validation of our prediction to be able to see if we could use this. Here, we went away from commercial farms because we really wanted to control a lot of the different things that were going on. So we took a research farm where different weed scientists, plant pathologists, soil fertility experts are planting crops. They're trying to do this as much in the way that commercial farms would be run for research purposes. And we were able to look at two different fields. We figured through our models that one of our fields, A, would have a higher predicted prevalence because it was closer to water. It was down towards the southern end of our farm, where you can see in our predictive risk maps, looking towards the southern area of this southern part of this farm, where it's red and green. This is where, red and orange. This is where our models, based on the different landscape and soil features, were pinpointing higher predicted prevalence versus B, which was up towards the north part of our field where it was blue. So we wanted to look and see what we found in terms of Listeria monocytogenes in these two spinach fields from planting to harvest. We wanted to identify factors, see if we could actually validate those prior models where we actually see what was correct. And we also wanted to look specifically at some of our weather factors, and as well, did we see a transfer to the produce, right? That's a huge question, right? We can certainly tell you where Listeria monocytogene is going to be in the soil, but would that actually transfer to the crop? So looking at this, we saw a pretty interesting distribution in our fields based on soil drag and leaf samples. We were able to see from our water samples that about 80% were positive for Listeria monocytogenes, which made this a really unique experiment since we were actually irrigating from those from that creek. What was interesting on this is that we assumed that the drag swab samples would be higher because they covered more surface area. But what we thought was really promising is that our leaf sample prevalence was very low. So we didn't see a lot of Listeria monocytogenes on our leaves, which showed that the transfer is quite low, less than 2% in this particular study, which was really great when you're starting to see sometimes prevalence is as high as 20%. When we looked at sort of what risk factors were influencing Listeria monocytogenes from being detected in these fields, we had noted that field A from our models would be higher in predicted prevalence. And we were actually able to see that field A was 2 times as likely as field B to find a Listeria monocytogenes positive. We were also able to see that rainfall within 3 days, we were 1.5 times more likely to find Listeria monocytogenes within a rainfall event. So this was nice because we were able to validate some of our model predictions in terms of picking out those fields or those locations that would be at higher risk. And then interestingly, we were able to see rainfall was really making a big difference. When we looked at the rainfall more specifically, here we have the predicted probability of Listeria monocytogenes, and you can see the more rainfall that you were getting, the higher predicted probability. And this mirrored in what we were seeing in our actual field data as well. Looking at field A specifically, this was our higher likelihood of finding Listeria monocytogenes. We really wanted to see were we seeing this again, where we saw spatial clustering or where it wasn't uniform. We were able to use some of these Ripley's K functions to show that we were getting the yellow-blue areas. They indicate Listeria density. So hotspots are in the green and yellow, which are around the edges, not in the center. We sampled these plots every 5 meters, so we had a really good distribution or uniform distribution of sampling. So if it was uniform in the field, we would expect to see a little bit more of a random pattern, but we weren't seeing that. We did a quadrant analysis, and this also through some conditional Monte Carlo simulations was able to show quite significantly that this result indicated evidence of Listeria monocytogenes positive soil samples were clustering in field A towards the edge. So we had edge effects that were going on. It was not random. Looking at our distribution of Listeria isolates, this also mirrored the same thing. We have our GIS-based image. We have our hotspot density plot. And again, we're able to see that on that edge, we're seeing all of those different Listeria pop up. We're actually also able to see too that the red dots share the same allelic type as what was coming from the creek. So somehow, even though we were distributing, this irrigation water uniformly in a field, we're getting these edge effects, whether that's based on low levels of the field or drainage. This is where we're starting to see the Listeria hotspots form towards the edges of this field. So kind of our summary and final thought is that these GIS-based models show a lot of promise in identifying trends and establishing measures of risk associated with contamination events. What was nice to see is that we were able, based on sampling efforts and prediction models, to go out into fields, whether commercial or research farms, and show that those predictions were actually working. When we thought we would find more Listeria, we were based on the computer models. And this is nice because it might yield a big return on an investment for food safety. You're going to have gaps that encompass the overall farm. But if you know that there's particular fields or particular locations on your farm where you may have a higher likelihood of Listeria monocytogenes or a pathogen, this is going to cause you to change some of your things? Can you do different crop rotations? Can you avoid that field at certain times of the year? Can you put in different mitigation, whether that be planting an extra row or two that you're not going to harvest near those edges, whether this can tie into laser leveling of fields for better drainage. There's many things to do if you can target an area or a problem area, especially if you can forecast different knowledge. Of course, it always takes a village. I'd like to thank my colleagues at Virginia Tech, Cornell, and Florida, as well as my laboratory group. I'd like to thank the funding sources, and a special thanks to the produce growers who participated in these studies. They were able to allow us and give us a lot of trust and freedom. And hopefully in the end, it was really interesting for them to find out sort of where on their fields, a lot of them had ideas of where they thought there might be more or less pathogens based on certain knowledges they had of the field, where the animal intrusion is, where low levels of the fields are, etc., different soil types, and seeing how these kind of interplayed with the models was really fun. So with that, thank you, and I'll take any questions.
Okay, thanks. Interesting to take a different look at this types of data as it relates to the spatial characterization. So any questions for Laura at this point? Please put them in the chat box, or it's a pretty small group. You probably unmute yourself and ask the question in person if you'd like. Just while we're waiting for some questions to get typed in, one thoughts I had was the edge of the field there was obviously trees, and it's closer to the source of the water and so forth. What was was there any observations on birds and whether they're related to being a source of the Listeria?
Yeah, we've looked at birds or avian populations for different foodborne pathogens, and with Listeria, they're not really a big source or contributor. That's more on the enteric pathogen side, so Salmonella and some of the Shiga toxin producing E. coli. We did collect quite a number of bird feces and animal feces, and we didn't really see a very high prevalence of Listeria in those samples. So I think with that particular field or some of the studies that we've been seeing, especially with Listeria, it's quite tied to some of the soil properties around drainage, some of the potential for water flooding or overcrusting, runoff, things to that nature, versus animal, which might be more on the enteric pathogen side.
Thanks. Again, I'm not seeing any questions come in, but I guess another one I'd ask. So realizing that UV light, sunshine has an impact on that, were those sides of the field such that they got more shading, again, because of being closer to the trees and so forth? Was there been any impact there?
Yeah, no, and that's actually a great observation. Those areas of the field were more shaded due to some of the trees. And we do see that in areas that can dry out faster. We will see less of a chance to find that. And it's very much tied to weather. You can see with some of the other Listeria models that, again, if we have increased rainfall where you get a lot of moisture, see higher prevalences. Also, if you get lower temperatures, cooler, shaded, it has a little bit better survival than if it's in the heat. So.
Okay. All right. Well, I haven't seen any additional questions come in. We'll encourage people to go ahead and pop them into the chat box, and maybe there'll be something there right after the break. But I think we'll go ahead and take our bio break now. We're about 10 minutes behind where we should be, and that's okay. Let's again come back at 10:30. So in about 9 to 10 minutes, and we'll start up again with the last 3 talks for this morning. So.
here under the food safety group talking about this because in particular, untreated raw animal manure.
Yeah, sorry, folks, I was muted there during the break. I'm going to restart this.
Hello, everyone. My name is Michelle J. Russell, and I am a research microbiologist at the Western Center for Food Safety at UC Davis, and I really appreciate the opportunity to be with you today. This is my first time recording a solo Zoom call, so hopefully this all works smoothly. I'm going to talk about food safety of biological soil amendments in organic and conventional agriculture. And this talk leans heavily on recent research, collaborative research that addresses some of what Dave Ingram talked about in terms of generating data to inform policy related to the produce safety rule, and specifically the subpart F that covers soil amendments. So the main highlights of my talk, or take-home message, if you fall asleep for the rest of it or need to go do something else, is first of all, untreated biological soil amendments of animal origin, or as we call them, BSAAO, are a potential source of zoonotic foodborne pathogens. And I'll explain what that means. E. coli, in our field trials, we have found that it dies off pretty quickly within 30 to 60 days after applying raw manure in conventional and organic soils, but there is a possibility it can regrow, especially following rainfall, and that can go out past the USDA National Organic Program's wait period standard of 90 or 120 days. So I'll show you some of that research. And then finally, root crops and vegetables grown close to the ground are at increased risk for transfer of E. coli from contaminated manure amended soil. And so these would be scenarios that are higher risk when considering use of these amendments. A little bit of background about the public health concern. I am here under the food safety group talking about this because in particular, untreated raw animal manure can pose significant microbial risks. And that can be from directly handling and applying it, but there can also be secondary issues such as storing manure near adjacent farms or applying it, having runoff, wind, or cross-contamination with equipment. So it's a sensitive product to have on the farm. What I mean by zoonotic pathogens, these are pathogens that go from animals to people. They can actually go the other direction too, but when we're talking about food safety, we're really looking at mostly bacteria that come from animals. And the biggies in produce food safety are E. coli 0157 and Salmonella in particular, which are shed in fecal material and often of ruminants and other animals. There are some others that are less common in the produce environment. Parasites are another risk factor, that another group of organisms that can be found in manures. You'll see that I didn't put viruses here, and that's because most of the viruses that are of food safety importance, especially in produce food safety, come from humans. And whereas these bacteria and a few parasites are mostly from animals. So you're not thinking about food workers, you're thinking about ultimately an animal source, including manure. The transmission cycle is, I don't want to point the finger at the cow, but the truth is that cattle are considered the primary reservoir of E. coli 0157. And I use this as an example because it has, although it's still relatively rare, it has had major impacts on our produce and beef economy, and huge efforts in the regulatory community to try to, and the industry to try to keep this bacteria out of our food supply. And in cattle, it doesn't make them sick. So you would be collecting their manure, and the cattle can look completely healthy. They can be on grass or they can be on grain. Either way, 0157 can be in these cattle, but it can vary with diet and season, but it's very unpredictable. And so in the end, you have to always think that a raw manure source, especially out of a ruminant, could contain a pathogen. In terms of have we actually seen outbreaks and recalls, we have. It's not a daily occurrence, certainly, and a lot of the produce outbreaks, it's not real clear the exact mechanism of how contamination happens, but for sure soil amendments are looked at during outbreaks. And there was a report in the literature about people getting infected from manured garden, home garden, and I do a lot of outreach with home gardeners that don't understand that horse manure and other manures can carry pathogens. There was also a relatively large outbreak that got traced back to salads. And when they did the investigation, it looked like a contributing factor was broadcast spraying of manure from an adjacent dairy that might have drifted over into the lettuce fields. And there has been a salmonella recall that where untreated chicken bones that were composted was suspected as a source. So this is a link to and just an aerial photo from that outbreak that did involve romaine lettuce traced to California, Central California. And it shows that the leafy green fields are in proximity to dairies and cattle forage crops. And this has generated a lot of talk about good neighbors and how we're careful when growing produce to also be in communication with the neighboring ag operations so that risk factors aren't introduced accidentally. So what type of animal manures are used as soil amendments in crop production specifically? It's actually all over the board. I've seen everything from what you'd expect, poultry, cattle, small ruminants, but also exotics, elephants, camels, rabbits, llamas. If you go on Craigslist, you could see any of these. And I have horses, and I've certainly used horse manure before around my non-edible landscape or composted it. So a couple of definitions that are important, and sometimes anything that's a pile of stacked manure is called compost, and it's not until it goes through a validated process of time and temperature. When we're talking about these BSAAOs, it's raw. It has not gone through a validated treatment or a composting process. And just leaving it stacked for a year, it's still raw manure. Time alone is not enough to make compost. And that is a misunderstanding when we've done surveys with various farmers, especially smaller growers that might be making their own compost or getting manures from neighbors. Untreated manure slurries. Here in California, we have large dairies. In other parts of the country, large swine operations where the manure liquid goes into a lagoon. There's also compost teas that might be made with raw manure. Those can be very risky because they're sprayed right on the plant leaves. And anything that's mixed with raw manure, if it's green waste mixed with raw manure, it's still raw manure and falls under the produce safety rule as Dr. Ingram probably talked about. I will point out also there's some non-manure based animal origin soil amendments that are considered safe to use because they've gone through a validated treatment process, but we still worry sometimes about them getting cross-contaminated, and one in particular at the bottom, they're heat-treated poultry pellets. We are doing some research on those because they seem to attract crows and other animals, and we don't quite know what might be going on, but you have to still consider all of your sources of soil amendments, even if they have gone through a treatment. But from this point forward, I'm going to just talk about raw manure and some of the research we've done, especially related to produce food safety. To begin with, we have done some surveys with FDA, with Substand Group, and for risk assessments on different types of manures. We've looked at foodborne pathogens, the E. coli and the salmonella in cattle and poultry manure, respectively. And this is a picture of a windrow composting operation where they're bringing in feedlot manure. And these are large-scale operations. This is a dairy in California where the lagoon, it goes into a pump, the liquids go into the lagoon, and then these dairy solids get stacked up, and then they can be brought out and put into windrows and also composted. But we do find that these have pathogens before they've gone through that proper composting process. But the large farms are not the only ones where we find pathogens. here's a couple pictures. I had a study on smaller farms and medium-sized farms, and it was very common that they would be diversified and have animals, and they'd also have crops, vegetables, roadside stands, go to the farmer's market, maybe sell to local restaurants. And so you might go to those farms and they would have stacked manures there and composting that we did find pathogens. They were not limited to the large dairies and feedlots and poultry operations at all. So it's a universal issue to consider. And then just one other, as far as it goes with different production practices, there's one that falls in between that we're doing some new exciting research on. Maybe we'll be back sometime. My colleague, Alma Perez, has been leading quite a bit of this, and it's looking at rotational grazing. And it's interesting because is it an animal intrusion? Is it application of raw manure? Is it both? Basically, they are fertilizing, during a either in orchards or in a fallow or stubble field, crop field. They'll utilize sheep or other animals to graze, and then on a cover crop, especially, and then rotate them off and plant their crops. We did do a preliminary study looking at E. coli die-off in an operation here in California, and I have the reference here in the slides if you want to read more about that. Some of our research has actually looked at who's using raw manure. It's not used a lot out here in California because of the Lethal Green Marketing Agreement and other buyer requirements that basically ban use of animal manures. But in some parts of the country, it is important, and especially to improve soil fertility and quality, and also to help manage manure in a region. And I'm sure there's a lot more about that at this conference. And so I'm kind of the one bringing the bad news, talking about some of the problems that can occur, but hopefully solutions too. We do have a paper that came out assessing who's using raw manure, specifically in the organic world, and it found that there are quite a few crops where raw manure is used around the country on produce for human consumption that might be eaten raw. So it falls under the produce safety rule, such as apples, lettuce, spinach, and so on. Our survey confirmed what we already knew from the organic community, is that, soil health, nutrient management, soil till cost, and waste management are all top reasons that raw manure is used. The sources, poultry and cattle led, but quite a bit of horse manure and as well as small ruminant manure being used. The big research question has been about these intervals. So the USDA National Organic Program, which is a marketing program, not a food safety program, but they do have in their standards a wait period from the time of putting untreated manure down to the time of harvesting. If the crop touches the ground, you have to wait 120 days. If the crop is raised, like tomatoes on a stake, then it's a 90-day wait period. But this wait period really had not been validated. And it is used in conventional too, in terms of good agriculture practices. So it's been a guide for farmers using these manures. And so we were funded to do research and look at whether or not these application intervals are really appropriate. We're thinking specifically about food safety. And one of the early collaborations did include Manan Sharma, who I believe is another speaker today. And we had a multi-regional study going on from the East Coast over here to the West Coast. And this was an early study, but we pretty much stayed with the same design in all locations, and with a few tweaks for regional differences. But basically, we're using these non-pathogenic indicator E. coli strains that we mix with manure, and we simulate a commercial field. And we've done tomatoes, spinach, lettuce, quite a number of different trials over the years. This first one was all the way back in 2014, and it was interesting. We found with these, we used horse, steer, goat, and chicken litter, as well as controls, and we found this steady die-off on the first 30 to 60 days. But then we had an interesting finding with a rain event, heavy rains, partway into this study, and of soil only. and had what we think was regrowth in this peak, and it did occur after the 120-day cutoff range. So this was just in soil. And, so we saw this nice log reduction in the beginning, and then this possible rebound with rain. And we didn't see a lot of differences between the manure types on longer survival and higher levels in the poultry manure. Another study to look for, and right now the International Association for Food Protection is going on the same week. And we have 4 different abstracts on this very large study. It's in its last year, funded by the Organic Ag Research and Extension Initiative. Alda Perez is the lead PI. I'm a co-PI. We've got PIs in Maine and at USDA ARS and in Minnesota, working also with the Produce Safety Alliance. So it's both outreach and research, and we've done quite a few field trials. And this is a poster that we have on Wednesday this week, also virtually, with some preliminary findings from my group on the field trials, where we looked at root crops and being carrots as well as spinach that was close to the ground and growing during our more fall winter season. And we did at a certified organic farm on campus. And we did find that we saw transfer, especially under rainy conditions, more than 120 days out. So for those particular crops, there is some concern, and we're right now analyzing regional data, trying to see whether we need a metric that is a number like that, or if there is something more nuanced to address the food safety concerns related to using raw manure, especially in these crops that have been more vulnerable to contamination with E. coli, pathogenic E. coli strains, and strains that come from animal manure in particular. So that's all that I have today. Thank you very much, and I'll be joining the Q&A to answer any questions. And I thank my lab and all of the collaborators for their help with this.
Okay, I'm going to try to do some Q&A now for Michelle's talk, although I don't think she was able to get on for the Q&A. I know that a number of these food safety folks had national conference this week, and I don't know that all of them were able to get on this morning. But are there any questions that the group might post that might be able to be answered by some of our other speakers? So give a moment here. Okay. So Laura has indicated if there are any questions for Dr. Michelle, that they will try to answer them. So please go ahead and post those. Okay, well, during the next talk, if you think of something you wanted to have asked Michelle, go ahead and post it in the chat box. I think we'll go ahead and move on to the next talk. So the next presentation is going to be a duo team from Montana. I'll give their perspective on safely utilizing animal manure and compost on small produce farms.
Hi, I'm Tommy Bass with Montana State University Extension, and I'm joined here today with my colleague from the Montana Department of Agriculture, Andrea Sarche. We're going to present our Montana perspective on safely utilizing animal manure and manure composts on small produce farms. We want everyone to take away today that the rules and best practices don't have to be overwhelming, and produce farms can safely benefit from using manure and compost. What are our manure sources here in Montana? Well, we're primarily a cow-calf state, though we do have some backgrounding in cattle finishing. we probably have about 40 or 50 large hog operations. And those small feedlots and backgrounding lots in the hog operations really represent the only manure in the state that can be collected, processed, composted, treated, etc. And all of our counties have sufficient conventional crop ground relative to the manure nutrients that are produced to agronomically assimilate those nutrients. Now, there can be some pockets of high nutrient concentration within a county or around certain operations, but this is usually easily remedied with nearby export and nutrient management planning. So where are our nutrients going right now? Well, manure and manure products are primarily going to conventional crop and forage ground. Somewhere second or third in there, it's going to reclamation projects, and that can be mine cleanup, construction and erosion and sediment control. Also sending some of these nutrients to acreages and organic cereal grains. Montana is a leader in those products nationally. And our sod farms and nurseries and greenhouses are also using manure-based nutrients for their enterprises. But the emerging opportunity and market for manure-based nutrients is our small produce farms and market gardens. And really about 8 years ago, I started getting more inquiries into my extension office from entrepreneurial vegetable and specialty crop producers looking to acquire manure and looking for advice on how to use manure and manure products. They're asking about renovating new acreage, new leases, new purchases, new farms. So wanting to improve their soils first and foremost. And then they were seeking information on how to use manure and manure products for their ongoing fertility. And in these images, we see some MSU students at our organic produce teaching farm applying and spreading a manure-based compost in a high tunnel greenhouse, and then incorporating that with the rototiller. And these hoop houses and high tunnels are pretty important for us. You see in the first picture that sunny and dry, and then in the second picture on the rototiller day, while a spring snowstorm has come through and you see the piles of snow outside. There seem to be some perceived barriers in connecting the animal ag sector with these specialty crop producers. And that means that we're having missed market opportunities for moving these manure and manure products between the sectors. And that just is going to have to be answered by Extension, Department of Ag, and the producer associations through facilitation and initiation. And what those barriers may be about is the fear and misunderstanding around rules and regulations. So concern over food safety and accidentally bringing in pathogens from the animal sector into the produce sector. Concerns over herbicide residues in compost or the manure itself, residues that have passed through the animals perhaps. And then concern over what the implications would be for those producers pursuing organic certification. And I want to real quick go over a case study of a farm that is incorporating all of these concepts that we're talking about here today. And Amalthea Organic Dairy is a small diversified farm and cheese plant. They have a dairy goat enterprise, pastured pig enterprise, as well as an organic vegetable enterprise. And all of those production areas, of course, produce organic residuals that can be used for their on-site compost manufacture. And they make that compost first and foremost to fertilize their feed crops. So some significant acreage and feed crop for the goats, for their vegetable enterprise. And then when there are surplus nutrients and compost available, they have occasional bulk and bagged compost sales. But basically they are moving these animal-based nutrients through all of their plant and animal enterprises in the closed loop. All right, I'm going to pass it over to Andrea now.
Right, so I just want to go through just talk through some key produce safety and organic standards for manure and compost use. I'll start by giving a very brief overview of the Food Safety Modernization Act and the Produce Safety Rule, and I'll dig a little bit deeper into some of the specific regulatory requirements, and then also talk through best practices and how that overlaps with the USDA GAP. With the regulatory requirements, you may hear me use the word must in relation to the regulatory language. of the produce safety rule and with some section symbols so that you can reference that later. And then with the best practices, I may use the word should to kind of differentiate those two things. And then we'll just talk more broadly about the implications for produce growers. So what is the Food Safety Modernization Act? FSMA, of course, marks a really big change to our nation's food supply, and it encompasses the entire food system. So there are 7 rules of FSMA, and the produce safety rule is just one of those rules that we're going to focus in on today. Focuses on prevention of food safety issues, and all the rules of FSMA are based. So it's important to know who is affected by FSMA, who's going to be required to comply with the rules of FSMA and the produce safety rule. We're talking about produce growers, but specifically anyone who grows, harvests, packs, or holds produce. So that is really key there, and I want to talk through briefly what the different statuses are in regards to the produce safety rule. So you may be exempt from the produce safety rule if on average your produce sales are less than $25,000 per year. And that means you don't have to comply with the rule at all. There will be no inspections or any other requirements there. Someone who is qualified exempt is a person who, a farm that has less than a total of $500,000 in produce sales. And in addition to that, most of those sales are local. So that is going to be the majority, or more than 50%, must be within the same state or 275 miles of the farm. And if you don't fit into either of those two categories, you may be covered by or subject to the produce safety rule and have to comply with all those requirements. So other folks that are possibly having to comply with the rule are wholesale food establishments and importers of produce. It does not, however, apply to retail food operations. And there I have the video in the lower right-hand corner there. We have a link to that at the end. That goes over in much more detail. It's about a 7-minute video, but it really helps people understand if that rule is going to apply to them. All right, so specific regulations on those soil amendments. First, we have to really define treated versus untreated soil amendments. A treated soil amendment is going to require a controlled process such as composting or heat treatment that will fully decompose that organic matter and reduce pathogens. Temperature is going to be the primary method of pathogen reduction, although chemical and biological are contribute to that reduction as well. So only a treatment process that's been scientifically validated will ensure pathogen reduction. We'll talk more about that a little bit more later. So process monitoring and record keeping are also going to be critical there. So for those untreated soil amendments, there is no minimum application interval for treated soil. So you can apply those all the way up until the day of harvest. However, for untreated soil amendments, we're talking about amendments that are considered higher risk since they have not been treated to reduce those pathogens. Some examples of untreated amendments might be raw manure, aged or stacked manure, agricultural teas, and then of course any soil amendment that has been mixed with manure or an untreated soil amendment. So the reason that, as Tony kind of mentioned too, the reason that those manures present a food safety risk is because they can spread and carry human pathogens. And then as far as the untreated application interval goes, the FDA has not specified an application interval yet, so the recommendation is to follow NOP standards, and that is 120 days for crops that do contact the soil and 90 days for crops that do not contact the soil. Okay, so as far as handling and storage goes on the rule requirements, so the musts would be covered farms have to handle, convey, and store soil amendments so that they don't become a potential source of contamination. They must not contaminate produce, contact surfaces, water sources, or any other soil amendments. As far as some shoulds, best practices go, designating specific equipment and tools for handling those soil amendments. You can use paint marking, for example, on your tools to make sure that you're only using those on those untreated soil amendments. Developing SOPs for cleaning and sanitizing equipment and tools, and also being thoughtful of where the traffic is going, directing your foot traffic and equipment traffic around those soil amendments to reduce that risk of cross-contamination is really helpful. Be thinking about runoff, leaching, and wind drift, and then keeping manure and finished compost in separate areas so they don't cross-contaminate. On the record keeping side of things, if you're doing on-farm composting, the requirements are to establish and keep records that document those process controls. So more specifically talking about that, we're looking at a scientifically validated process, which really documents the times, temperatures, and turnings that you're using to make sure that would comply. And if you are purchasing any treated biological soil amendments, you must keep on hand that documentation that talks about what process controls they use and that's sufficient for that. Best practices, you can also look at the type and source of the soil amendment, rates and dates of application. Those are good things to do as well, not necessarily required by the produce safety bill. All right, so as far as the overlap between GAPs, USDA GAPs and and the produce safety rule, these are more best practices. GAP standards are very similar in terms of handling storage and record keeping. But as far as selection goes, GAP standards going above and beyond that would be to just stay away from those untreated soil amendments. That certainly would reduce your risk. Look at the application timing in terms of you can extend it beyond those NLP standards that we talked about. And then application methods, just making sure you're not putting any adjacent crops at risk as well. So getting into the organic side of things, we have an organic system plan example form here that kind of leads you through a bit of a flow chart to figure out what your risks are and how to manage those. So this is just an example of that. And more specifically, in a couple areas for organic certifications, you really need to be thoughtful of be looking on the organic approved products list to see if you are currently using any prohibited materials or soil amendments. And then you have to go 3 years without using those in order to transition over to organic and start your certification process. As far as ongoing use, we already talked about that 90-120-day rule. So that applies all the time for same thing as far as untreated and treated soil amendments, manure versus those other treated ones. And then ag chemical residues, there's a number of things. While the NOP standard doesn't specifically address ag chemical residues, producers can take a number of measures to be sure that they really won't have an issue there in terms of getting those letters of certification and analysis from anyone purchasing those amendments from as well.
All right, thank you so much, Andrea. So in summary, manure and compost are valuable nutrient inputs for small produce farms. The food safety risks associated with using manure can be minimized with proper management, whether complying fully with the FSMA related produce rules or following the recommended good agricultural practices. It's also important to remember manure and compost products are not generally prohibited. Best practices and application interval rules and whatnot should be followed, but using the GAPs, following the rules will allow you to bring manure and compost onto a produce operation. Here are the contacts for myself and Andrea, both phone and email, and a copy of the link that was referenced in slide 11, the Am I Covered by FSMA 7-minute video. With that, we thank you for your time, and we look forward to the live question and answer session. Thank you so much.
Okay, so Tommy and Andrea are on today and are available to take questions. Tommy has entered into the chat box some of these important fact sheets and also a YouTube link for a video. So if you want to pop any questions in the chat box, we can take those for a couple minutes. We've got about 3 minutes here yet before we'd start up our next presentation. Any additional thoughts, Tommy or Andrea, that you have, having listened to your talk this morning, that other points you would like to make or emphasize?
Well, I think one thing I noted in the chat box, I'm just looking forward to reading some of that research that was presented and the references provided most recent previous to our talk. As of yet, from what we've seen in Montana, we've had a couple little blow-ups with broadleaf herbicide residues coming through compost imported to these types of operations. But we have not had any sort of significant documented human health issue yet. Yeah, not much to add at the moment.
Okay. Anything on your part, Andrea?
The only thing I can think of, and Tommy mentioned it as well in his presentation, but just being mindful of, grazing animals, and that being also an application of raw manure. I didn't really bring that up in the presentation, but that's probably fairly obvious to folks. Just want to make sure that people are thinking about that in rotation.
Okay. And that came up a little bit in Michelle's talk from California, where they had grazing within small orchards and so forth. So good example of that. Okay, any other questions that folks have? And again, we're a small group, so if people want to just go off mute and ask a question, feel free to do that. If you don't want to put it in the chat box. So, okay, well, thank you, Tommy and Andrea, and go ahead and move on to our last presentation for this session. And that talk will be looking at electrochemical disinfection of irrigation water using a flow cell.
Hi, my name is Geletu Chen. In this presentation, I would like to talk about our results on the electrochemical disinfection of irrigation water using low-cost graphite or titanium electrodes. This method is effective for disinfecting water containing low chloride concentrations by utilizing naturally occurring chloride ions. Therefore, chemical disinfectants are not necessary, and it also produces less toxic disinfection byproducts compared to the conventional methods using chlorine gas or hypochlorites. Foodborne diseases caused by pathogens are a significant public health burden. For example, there are about 48 million cases of foodborne illness and 3,000 deaths each year in the United States. And the associated expenditures on medical care and lost wages are more than $15.6 billion. And it is also costing the food industry billions of dollars in the recalls of tainted foods, lost sales, and legal expenses. Therefore, the FDA Food Safety Modernization Act, which went into effect in January 2016, aims to shift the focus of federal regulators from responding to contamination to preventing it. Importantly, the U.S. irrigated area reached 55.7 million acres, and irrigation water is the most important route of pathogenic contamination in fresh produce. Therefore, the disinfection irrigation water is a practical approach to preventing foodborne illness. Disinfection of irrigation water can be achieved by chemical or non-chemical methods. Most chemical disinfectants are hazardous to produce and transport, and their use also leads to the accumulation of chemical in the soil, water, and the irrigated plants. Hypochlorites and chlorine gas are the disinfectant of choice for most applications because they are highly efficient and also provide low cost. However, their use leads to the formation of toxic byproducts, for example, chloride and trihalomethanes. Ultraviolet irradiation is a non-chemical method. It is, however, sensitive to turbidity, and it has no residual disinfection effect. Electrochemical techniques can enable the in situ generation of disinfectants such as chlorine and hydrogen peroxide. by utilizing naturally occurring ions and other species in the water. It can also enable the direct electron transfer between the electrode and pathogens. Therefore, on-demand disinfection is possible, and it can also be powered with renewable electricity, which is very important for remote and off-grid farms. The first part of this presentation is the disinfection of irrigation water in Hawaii using a graphite electrode flow cell. The flow cell consists of two graphite plate electrodes placed in parallel, and the water flows through the channel between them. The flow cell is connected to a bottle, and the water is circulated using a pump. This table shows the chemical composition of the two types of irrigation water, bottom well pond and open ditch. The bottom well pond water contains higher concentration of chloride, but it is less than 7 ppm. And the bottom well pond water also contains higher concentration of ammonia, nitrogen, and total organic carbon. This slide shows the effect of current density and flow rate on the disinfection of bottom well pond water. The flow cell was operated at open circuit for 60 minutes, and no disinfection was observed. In the first experiment, the red colonies on the plates correspond to coliform bacteria naturally occurring in the water, and it was completely disinfected after 10 minutes of treatment. In the following 3 experiments, E. coli was added, and it was disinfected after 20 minutes of treatment. However, the effect of current density on the disinfection efficiency was not quite clear, probably because the initial concentration of E. coli in the solution changed significantly. In the last experiment, there was a significant decrease in the disinfection efficiency, probably because at a higher flow rate, it became easier for the disinfectants produced at the electrode to react with organic matter in the bulk solution in the bottle. This slide shows the effect of current density and flow rate on the disinfection of open ditch water. The results are similar, but the disinfection process is faster, most likely because the open ditch water contains a lower concentration of organic matter. Chlorine and hydrogen peroxide are the most possible disinfectants electrochemically generated at the electrodes. In the bottom well pond water, the concentration of total chlorine increased with time, but free chlorine was not detected in the solution throughout the experiment. In the open ditch water, the concentration of total chlorine also increased with time, and free chlorine was detected in the solution at the end of the experiment. Apparently, the high concentration of ammonia and organic matter in the bottom well pond water consumed all free chlorine electrogenerated at the anode. Hydrogen peroxide was not detected in both experiments, and the concentration was confirmed to be significantly lower than 1 ppm. In addition, experiments in synthetic water without chloride showed negligible disinfection performance. These results demonstrate that the disinfection mechanism was controlled by chlorine species electrogenerated at the anode. To further confirm the role of free chlorine, a free chlorine solution was added to the bottom well pond and open ditch water. The free chlorine solution was prepared on-site by electrolyzing sodium chloride solution. The complete disinfection of the bottom well pond water requires approximately 0.5 ppm of free chlorine, but the open ditch water requires only 0.1 ppm of free chlorine. This difference is because the bottom well pond water contains higher concentration of ammonia and organic matter. The disinfection effect of hydrogen peroxide was further examined by adding a commercial hydrogen peroxide solution to the bottom well pond and open ditch irrigation water. The complete disinfection of the bottom well pond water requires approximately 200 ppm of hydrogen peroxide, and that of the open ditch water requires approximately 50 ppm of hydrogen peroxide. Therefore, the disinfection effects of hydrogen peroxide during the electrochemical treatment should be negligible because the concentration did not exceed 1 ppm. The electrochemical disinfection of irrigation water using graphite electrodes provides several advantages. First, the graphite electrodes provide low cost. Second, the production and transportation of highly reactive chemical disinfectants are not necessary. Finally, the addition of other chemicals is also not needed. However, the graphite electrodes undergo corrosion during electrochemical treatment due to the electrochemical oxidation of carbon. Therefore, we perform the disinfection of open ditch water using titanium electrodes. Titanium electrodes are well known for their high chemical and electrochemical stability, The experiments were performed in a batch cell assembled with titanium foils at both anode and cathode. Polarity reversing direct current was applied between the two electrodes to mitigate the formation of oxide on the electrode surface. In this work, the direction of the current was reversed every 5, 10, 30, or 60 seconds. This slide shows the effect of current density on the disinfection efficiency. The half period of the polarity reversing direct current was fixed at 30 seconds. The current density was first increased from 0.5 to 2, and then the experiment at 0.5 was repeated to confirm reproducibility. Finally, the experiment was performed at open circuit. No disinfection was observed at open circuit and when the current density was 0.5. When the current density increased from 1 to 2, there was a significant increase in the disinfection efficiency. The concentration of free chlorine and total chlorine in the solution was measured during the disinfection experiment. Free chlorine was not measured in the solution at all current densities. Total chlorine was not detected in the solution at open circuit or when the current density was 0.5. However, total chlorine was detected in the solution at higher current densities, and its concentration increased with both time and the applied current density. This slide shows the effect of the half period of the polarity reversing direct current on the disinfection efficiency. The current density was fixed at 2. The half period was varied from 5 seconds to 30 seconds, 10 seconds, and 60 seconds. And each experiment was repeated twice to confirm reproducibility. All experiments showed near complete disinfection of E. coli after 10 minutes of treatment. However, the experiment at 10 seconds showed exceptionally promising results, achieving the complete disinfection of E. coli within 5 minutes. The concentration of free chlorine and total chlorine in the solution was measured during the disinfection experiment. Free chlorine was detected in the solution only when the half period of the polarity reversing direct current was 10 seconds, and its concentration increased from the first experiment to the second experiment. Total chlorine was measured in all experiments. However, its concentration at 10 seconds was higher than other experiments. The trends in the concentration of free chlorine and total chlorine matched very well with those of the disinfection performance. In addition, hydrogen peroxide was not detected in the solution at the best performing conditions. Therefore, the disinfection mechanism should be due to the formation of chlorine species at the titanium electrode. The effect of chloride on the disinfection performance was further studied by performing disinfection experiments in synthetic water with or without magnesium chloride. Magnesium chloride is the sole source of chloride in the synthetic water. In another experiment, the magnesium chloride was replaced with magnesium sulfate to check the effect of magnesium ions on the disinfection performance. The results show that fast disinfection was achieved only when chloride was present in the synthetic water, and free chlorine and total chlorine were detected in the solution only when chloride was present in the solution. These results demonstrate that the fast disinfection was due to the generation of chlorine species from the oxidation of chloride ions at the titanium electrodes. An important question is why the titanium electrodes showed the highest disinfection performance when the half period of the polarity reversing direct current was 10 seconds. A possible reason is proposed based on the cell wattage data. When the half period was 5 seconds or 10 seconds, there was a fast increase in the cell wattage, which is probably related to the formation of an oxide layer on the electrode surface. At 10 seconds, there was a plateau after the initial increase in the cell wattage. It is expected that the titanium electrode showed high activity toward chloride oxidation during this phase. When the half period was 30 seconds or 60 seconds, the cell voltage increased slowly in each half period. A possible reason is that when the direct current was applied for a longer time, a thick layer of oxide was formed on the electrode surface, and the resulting electrodes showed low activity toward chloride oxidation. The suitability of the titanium electrode was examined by performing 30 tests at the best performing conditions. The odd numbers experiments lasted about 10 minutes, while the even numbered experiment lasted about 5 minutes. These results show that the complete disinfection of E. coli was achieved in each experiment. The cell voltage was also extremely stable during the 30 experiments, including the odd number experiments lasting for 10 minutes and the even number experiments lasting for 5 minutes. This slide shows the concentration of total chloride after the 30 repeated experiments. The first 15 experiments were performed on the first day, while the other 15 experiments were performed on the second day. On each day, the 15 experiments were separated into 3 groups, with each group containing 5 experiments. The titanium electrodes were exposed to air for less than 10 minutes between experiments within each group, but it was exposed to air for approximately 45 minutes between different groups. This result shows that the concentration of total chlorine increased in each group. It indicates that titanium electrodes become more active toward chloride oxidation during repeated experiments. However, after exposing to air for approximately 45 minutes, the activity of the titanium electrodes returned to the original point. This result indicates that the titanium electrodes were oxidized by oxygen in the air and lost the highly active surface state. However, the highly active surface state can be restored by applying the polarity reversing direct current. The titanium electrodes provide multiple advantages, including low cost, no additional chemical disinfectants, no chemical addition, and high durability. The concentration of toxic disinfection byproducts was measured during the experiment. Chloride was not detected in the solution, and the upper limit was determined at 0.1 ppm. Bromodichloromethane, dibromochloromethane, and bromoform were not detected in the solution. Chloroform was not detected at 5 minutes, and its concentration was 2.5 ppb at 10 minutes, and its concentration increased to 9 ppb at 20 minutes. However, these values are significantly lower than the recommendations for drinking water. As a conclusion, both graphite and titanium electrodes demonstrate disinfection of irrigation water, and the disinfection mechanism was controlled by chloride oxidation to chlorine. In addition, the titanium electrodes are expected to offer long-term stability. The electrochemical approach eliminates the addition of chemical disinfectants and also leads to the formation of low concentrations of toxic disinfection byproducts. We gratefully acknowledge financial support for this work from the Hawaii Farm Bureau and the Hawaii State Department of Agriculture. Thank you very much for your attention.
Okay, so very detailed bunch of electrochemistry there. Thanks for that presentation to round up our food safety section this morning. We've got about 14 people left on this morning. So 12 others minus Gallatin and myself. So are there any questions from the group of 12? I guess I'd ask one, maybe again, folks can unmute themselves if they want to ask in person rather than put in the chat box, but scalability of this, how do you see being able to scale this up, and have you done any work at larger scale than what was presented today?
Larger scale have not done it. So this is just the lab scale, very small batch cell studies. I think the titanium electrode is more scalable because it is very stable, but further studies have not been done it.
Okay, so there's one question did come in. Would a more inert electrode material such as PD or PT be better than the other electrodes? So
I tried platinum, so that result is not shown here today. I compared platinum coated titanium and titanium. So platinum coated titanium is more active, electrochemically more active, and it is more active for chloride oxidation. But our comparison shows that although platinum titanium is more active, but when it is applied for water with very low concentration of chloride, it needs a higher current density to oxidize chloride, because the platinum is also active for oxidizing water and organic matter. So it just needs higher current density to achieve similar chloride oxidation performance. So it is more active, but it requires more energy. So, and for the stability of titanium platinum, for application in water, the direction of the current has to be reversed to eliminate the formation of scales on the cathode. So during this kind of reversing the current density, the platinum layer is easily detached from the platinum layers easily detached from the titanium supporting layer. And we didn't do long-term study, but previous study shows that it has a lifetime of around 1 year or something.
Okay, great. Okay, any other questions that come in? Not seeing any. Well, thanks for that presentation. Looks like some promising technology there.
Thank you.
What I'm going to do at this point is launch the poll. If you guys would answer those 3 questions, I'd appreciate that. And then I will put up the QR code at the end, and we'll conclude our session for today.